So it's my really great pleasure to introduce Kentaro Tamura from the Shizuoka University in Japan. So Kentaro has a long-standing interest in the nuclear morphology, and he will tell us more today about the functional and spatial coordination between cell and nuclear shape in plants. So Kentaro, it's over to you.
Thank you very much, Christoph, for a kind introduction. I'm Kentaro Tamura from the University of Shizuoka, Japan. First of all, I really appreciate the organizers, David, for this session. I'm very pleased to be part of this meeting and have the opportunity to get back to SUB as a virtual conference. Today, I'd like to talk about the mutants we recently isolated, which shows abnormal nuclear and cellular shape as well. As you might know, plant nuclei have various shapes dependent on tissues. If you have a look at Arabidopsis seedlings, you would see many types of nuclear and cellular shape. For example, in epidermal cells of cotyledon, nuclei have spindle-like shape in the puzzle-shaped cells. On the contrary, in gourd cells, nuclei are smaller and more spherical shape than epidermal cells. In root tips, where meristematic cells actively divided, the nuclei are small. However, after cell division, cells begin to grow and the nuclei get elongated. So our question is how nuclear identity is maintained and what molecules are involved in nuclear structure in plants. To answer these questions, we have performing forward genetic approach to isolate the nuclear mutants and to identify the genes controlling the nuclear dynamics. Firstly, we generate transgenic plants expressing the nucleus localized GFP, and then mutagenize the seeds of these transgenic plants. Afterwards, under fluorescence microscope, we have isolated nuclear mutants, which show abnormal nuclear structure or impaired nuclear dynamics or movement, and named these mutants as kaku mutants. Kaku means nucleus in Japanese. These are proteins identified by our mutant screening. They are all localized in nuclear envelope. Kaku1 is plant-specific myosin 11I, and we found that this myosin controls nuclear movement and shape. Kaku2 is allelic to CLAN1, which is suggested to be a component of plant lamin-like structure. Kaku4 is novel type of nuclear protein, as Christoph introduced, which is conserved in only higher seed plants. So if you have a mutation in either CLAN1 or Kaku4, you would get smaller and more spherical shape of nucleus. Kaku4 shares the function with CLAN1, and also Kaku4 interact with CLAN1. So we therefore suggest that Kaku4 is also component of plant lamin-like structure and works together with CLAN1. Kaku3, which has not yet published, but it is a nuclear porin, which is a component of nuclear pore complex. Interestingly, this protein is required for nuclear anchoring at particular cell position against flow of cytoplasmic streaming. Today, I would like to talk about new nuclear mutants, Kaku5 and Kaku6. Okay, these are fluorescent nuclei visualized by DAPI staining in cotyledon epidermal cells of wild type and Kaku6 mutant. The phenotype of Kaku6 mutant was very different from previous Kaku mutants. Kaku6 mutant shows bigger and elongated nuclei compared with wild type. Also, we found that Kaku6 mutant is a dominant mutant. The phenotypes can be observed in both heterozygous and homozygous mutants, suggesting that Kaku6 mutation gives either a gain of function or loss of function. In addition to nuclear phenotype, CAC6 mutant has bigger size of cells. These are fluorescent images of cell membrane in leaf pavement cells of wild type and CAC6 mutant. Yellow highlight indicates one pavement cells. CAC6 mutant has bigger cell size and more complex surface pattern compared with wild type. We quantified the cell areas and found that CAC6 mutant has significantly higher number of larger cells compared with wild type. And CAC6 also has bigger organ size. These are mature leaves just before flowering stage. All leaves of CAC6 mutants have wider and larger areas, and leaf teeth tissues is highly developed in the mutants. Also in flower and dry seed, CAC6 mutation led to bigger organ size. We quantified the seed weight and revealed that CAC6 seeds have higher weight compared with wild type. These results suggest that CAC6 is required for nuclear and cellular size and controlling organ size. The trichome branching is also affected by CAC6 mutation. In wild type, it is known that more than 70% of trichome has 3 branches, and we cannot detect more than 5 branches in the wild type. However, more than 40% of CAC6 mutant trichome has 5 branches, and 30% has 4 branches. The 3 normal branches of trichome are just 20% in the mutant, suggesting that CAC6 has a function involved in trichome morphogenesis, in addition to nuclear and cellular phenotype. Based on these results, that CAC6 mutant has increased the nuclear and cellular area, organ size, number of trichome branches, we next examine the nuclear ploidy level with flow cytometry. In wild type, majority of cells have lower ploidy, such as 2C and 4C. A number of higher ploidy cells increased as plant grows. On the contrary, in CAC6 mutant, more than 40% of cells has higher ploidy level, such as 16C and 32C, suggesting that CAC6 dominant mutation led to increasement of nuclear ploidy, subsequently increasement of nuclear and cellular size. We also found that CAC6 dominant mutation affects reproductive growth. When we visualized the seeds in the slicks, we found that there are some spaces without any seeds, indicated by yellow arrows. And these are scanning electron micrograms of pollen grains. Compared with wild type, the mutant pollen grains have irregular shape, and sometimes collapsed pollen can be found. The FDA staining of pollen grain indicate that viability of pollen grain in mutant is significantly decreased compared with wild type, suggesting that CAC6 is important for development of male gametophyte. We have not yet checked the female gametophyte of the mutant, but we think at least impaired development of pollen in the mutant affects seed production finally. So what is a responsible gene for the CAC6 dominant mutant? A map-based cloning revealed that the CAC6 mutant has a point mutation in ECT1 gene, ECT1 gene. ECT stands for evolutionary conserved C-terminal region because it has a conserved domain called as YTH domain at C-terminal. In addition to this YTH domain, we found that unknown conserved domain at N-terminal indicated by yellow box is found in this protein. KAK6 mutation was found in this unknown conserved domain, and this mutation changes from glycine to arginine. The YTH domain is well conserved in eukaryotic cells, and it is known to function as methylated messenger RNA reader proteins to control its translation. This is a phylogenetic tree of YTH domain proteins. Green is Arabidopsis, violet is human, and brown is yeast. Arabidopsis has more than 12 YTH domain proteins, including 10 ectogenes here. Ect1 is here and in the Ect1-24 subclade. This is a sequence alignment between Ect1, 2, 3, and 4. The glycine residue mutated in the KAK6 is fully conserved in these ectoproteins, indicating the functional importance of this residue. We have isolated each ect knockout mutant and analyzed the trichome branching. As I showed you in the earlier slide, KAK6 dominant mutant shows increased number of trichome branching. However, ect single knockout mutant and double mutant with ect1 shows no obvious phenotype. They are similar phenotype to wild type. On the contrary, other group has already reported that ect2, 3, and 4 multiple mutants shows increased branching, like here, suggesting that ectoproteins have overlap function. And I don't show data here, but ploidy level phenotype is also similar to this. Only ect multiple mutant shows the higher ploidy phenotypes. These results suggest that KAK6 dominant mutation mimics ect multiple knockout mutant, and ect proteins are involved in nuclear ploidy. Okay, we next examine the ect1 localization with GFP fusion. We have generated transgenic plants expressing wild-type ect1 GFP and ect1 with a mutation glycine to arginine under control of native promoter. But we cannot distinguish the fluorescent pattern between wild-type ect1 and mutated ect1 GFP. They are similarly expressed in meristematic cells like shoot apical meristems, root tips, and lateral root, and they also localized in cytosol, but not in nucleus. Under normal growth condition, the fluorescence of ect1 GFP is dispersed in the cytosol. However, when plants are exposed to higher temperature for 30 minutes, both ect1 GFP forms aggregates in the cytosol like this. And these aggregates are co-localized with PAB2 MRP, which is a stress granule marker. Stress granules is known to have various types of translational factors and to control the protein translation in response to stress. Although we have no idea how heat stress is involved in ploidy control, but ect1 could be required for stress responses. This is a working model of KAK6 ect1 function. Ect1 is YTH domain protein, and YTH domain is known to interact with specific messenger RNA molecules and control the translation. So we think that Arabidopsis Ect1 could be involved in controlling the translation of ploidy-related genes, resulting in regulation of proper nuclear and cellular size. Okay, we move to another nuclear mutant, KAK5. These are fluorescent image of cell membrane and nuclei in wild type and KAK5 mutant. Originally, KAK5 mutant was isolated as a nuclear mutant, which has smaller nucleus compared to wild type. But afterwards, we noticed that KAK5 mutant has abnormal cellular shape in leaf pavement cells. So we firstly quantified both nuclear and cellular area of wild-type and KAK5 mutants. As shown here, KAK5 has significantly smaller nuclear area and cellular area too, compared to wild-type. We also found that pavement cell shape is different from wild-type. The leaf pavement cell has a very characteristic puzzle shape. They are neck domain and lobe domains. To evaluate cellular shape, we quantified lobe numbers. As shown here, KAK5 mutant has lower number of lobes in pavement cells, suggesting that KAK5 mutation affects both cellular and nuclear shape. KAK5 mutants show very pleiotropic phenotypes of plant growth. The growth rate of seedling in KAK5 mutant is very slow compared to wild type. After flower development, KAK5 showed dwarfed phenotypes, and KAK5 has smaller number of mature leaves. It suggests that early flowering phenotypes of this mutant. The length of rosette leaves is much shorter than wild type, but width is similar to wild type, suggesting that KAK5 is involved in leaf morphology as well. A map-based cloning revealed that KAK5 has a point mutation in spike1 gene, which encodes a guanine nucleotide exchange factor, an activator for small GTPase. We isolated several knockout mutant lines of spike1 gene and visualized the cell membrane. As shown here, all spike1 knockout mutants show abnormal cellular shape. We also quantified cellular area, lobe number of pavement cells, and nuclear area. All mutants show similar phenotypes: smaller cellular area, decreased number of lobe, and smaller nuclear area, indicating that spike1 is responsible for KAK5 mutant phenotype. To investigate cellular localization of spike1, we introduced the spike1 GFP under control of spike1 promoter into spike1 mutant spike1-8. We confirmed that spike1 GFP restores root growth in the mutant. Also, cellular area and lobe numbers are partially recovered by spike1 GFP fusions. These are fluorescent images of spike1 GFP. GFP fluorescence is dispersed in cytosol in all tissues we checked, suggesting that spike1 is cytosolic protein. The spike1 is known to be important for cellular shape, and it is well studied by other groups. Based on the previous reports, spike1 functions as guanine nucleotide exchange factor and activates small GTPase Rho, which is a row of plant proteins. The activated Rho proteins interact with other protein complexes and regulate cytoskeleton for cellular shape. So we think such regulation of cytoskeleton could be important for nuclear shape as well. It has been reported that spike1 activates Arabidopsis LOP2, 4, and LOP6, which are mainly function in leaf cells. So we examined the nuclear and cellular shape in LOP2-6 double mutant and LOP2-4, LOP6 triple mutants. As shown here, we found both cellular and nuclear areas are significantly reduced in these mutants, as seen in KAK5 mutants, suggesting LOP2, 4, and 6 and spike1 work together to regulate both nuclear and cellular shapes. Finally, we visualize the cytoskeleton and actin filament and microtubules and quantified their structure. When you see actin filament, CAC5 looks normal. The average angle and the parallelism of actin in the mutant is very similar to wild type. However, when you visualize the microtubule structure, the mutant has less microtubule compared to wild type. This heat map shows fluorescence intensity of microtubules, clearly showing that CAC5 has lower fluorescence intensity of microtubules. The quantification data indicates that CAC5 mutation affect microtubule distribution in leaf cells, suggesting that spike1 is required for microtubule distribution in pavement cells. Taken together, we made a working model of spike1. Spike1 encodes a guanine nucleotide exchange factor for activating LOP GTPase. In Arabidopsis leaves, mainly LOP2, 4, and 6 are target proteins for regulation of cytoskeleton. In our model, a microtube distribution in this cell is controlled by LOP2, 4, and 6. And this microtube distribution subsequently regulates both nuclear and cellular shape for supporting plant growth. But there are missing links between microtubules and nuclear shape. We still don't know what molecules are involved in linking microtubules and nuclear shape. Lastly, I'd like to thank all collaborators and the grant foundations. Of course, thank you very much for your kind attention. Thank you very much.
So thank you, Kentaro, for your nice presentation. So I don't know if we have questions. So maybe I have one question for you, Kentaro. So you found now many of these mutants affecting the nuclear morphology or the nuclear size. Did you notice in your screen any mutant where the correlation between the nuclear size and the cell size would not be tightly correlated or uncorrelated? I mean.
I think they are very strictly correlated. If you have a bigger cellular size, you would have a bigger nucleus. So I think the size correlation, there's some correlation between nuclear and cell size in Arabidopsis.
Okay. So I'm sorry, I can't see questions. Maybe I'm not looking at the good place. So if people have questions, do not hesitate to use the chat on the left hand of a panel. I can't see some. Hello, David.
Hi, no, I can't see any questions in the live now panel either. Kentaro, congratulations. That again is excellent results and some really significant progress being made once again, and that opens out new systems for us to investigate, So I think everything you find is leading on to connections, but also some really interesting future work. So yeah, congratulations, excellent.
Thank you very much, David.
And regarding, so maybe one more question regarding the CAC6 mutant. So you found this domain, so YTH domain at the C-terminus of your protein, and so it's a well-conserved domain. And if I understood well, it's something related to the methylated mRNA readers. So do you have any guess about the possible function of CAC4 in relation to this possible function?
You mean is there any link between CAC4 and CAC6?
No. I mean between this in CAC6.
Okay.
If this yes.
Well, actually, we have no idea what molecules are target for CAC6, but we are now doing RNA sequence analysis to identify the methylated messenger RNA, which can be target for CAC6 protein. But so far, we have no idea what molecules are target for CAC6.
And you have any idea if this protein is able to bind some RNA?
Well, actually, in vitro experiment, CAC6 can interact any methylated messenger RNA. So I think there's low specificity for, binding.
I see. Okay. So I don't know if there is no more question. Maybe we can join the next lobby for the virtual conference. It's the poster presentation, I think.
Yeah.
So
I think there will just be a few minutes before that opens for most people, but for you, Christoph, and for me, probably it opens quite soon, so we can go over. But please do go and visit the poster session that's related to InDepth, and we'll see you there in a few minutes. Great.
Thank you again, Kentaro, for your presentation. Yeah, that was
Thank
nice.
you. Thank you very much. Thank you.